Okay, so hi everyone. Pleasure to be here, and thank you again for the invitation. I'm Shai. I'm based at the Technion, and actually wonderful to come after Yoni. He probably saved me some slides. I guess for disclosures, I'm also the co-CSO of the Human Immunome Project, which is a global nonprofit, and the chief scientist of Cytoreason, a pharma AI company. And I also head the Technion Healthy Aging Institute. We'll touch on that a bit later. Okay, so I really kind of what my research is around is studying immune system variation. And the way I think about the immune system is really like a sixth sense. It's basically the system in our body that's in charge of maintaining homeostasis of everything else, right? And to do so, it basically keeps monitoring internal and external environment and adapting to it. So often we say immune homeostasis, which is a misnomer, because really, if you think about the immune system, this is a system that's in constant allostasis. It keeps changing over time. And therefore, the way to study it is over time on different time scales. You can think about immune response, you can think about disease progression, and you can think about a longer process of immune aging. So the way we've kind of been tackling this is really kind of studying this complex system at high resolution over time, right? Measuring everything that we can about it. We strongly believe we should be studying it in humans because they live in the real world. It's both more relevant from an impact perspective, but also more correct, we believe, for studying this system, which is all about kind of getting exposure. And with this, so we measure the system over time. We measure it in high resolution. We measure it in a multimodal fashion. And we perform functional assays to understand how changes in the immune system ultimately change over time. Back in 2019, we published a paper in Nature Medicine. This is a collaboration with Mark Davis, where I did my postdoc, where in 2007, we started studying healthy humans and how they change over time at various ages. Basically, this is called today the cellular cohort. It's the longest running high resolution study of the immune system. And we collect samples annually from individuals, and we vaccinate them with the seasonal flu vaccine and COVID and so forth. And we test to see how things actually change over time with the latest multi-omics. Just to give you a sense, if you look here, over here, what you'll see is that the classic hallmark of immune senescence is the shrinking of the thymus and the drop in naive CD8 T cell frequency. That's the black line here is the average. Okay, that's how the entire cohort behaves. Thousands of papers published that naive CD8 T cells drop with age. But because our cohort is longitudinal, we can actually derive individual slopes for naive CD8 T cell frequency, and you can see that they're all over the map. Basically, the average doesn't represent anyone. And in fact, there are people who actually go against the trend. This is not just true for naive CD8 T cells. Just to give you a sense of the resolution of this cohort, we're measuring in blood 120 different cell types, this well-described cell types, of which 79 are basically reciprocal to one another, so a very broad range. And this is true for over 50% of the cells that have these types of behaviors. They change with age, and they change drastically in different ways in different individuals, and certainly at different rates. So having this high resolution individuals, I can take everybody here in the room, measure your blood cell frequencies, and describe you just as a vector of immune cells. And if I do so, I can start deriving how that vector changes over time. And this is what we did back in this paper in 2019. We described for the first time a trajectory of immune aging. This is you can think about this as people like to think it's a biological clock, but in fact, there was no chronological age going into this data. Basically, all we did is follow people over time. It's a developmental process that we're trying to map in humans and keeps happening to us as we live life. Oops. Okay, so that's what you see here on the left-hand side, where every point is one individual in our time course, at a single time point, and the color denotes the chronological age, and the position basically describes this vector of immune cells, which is just basically data-driven trajectory building by a machine learning algorithm. Every individual on this cohort that is old has a positive slope, so it's moving along this trajectory. You can think about this as a pseudo-time, so it doesn't describe chronological time passing, it describes some other process, so it moves to right from left. Okay, and every individual who's old has a positive slope on this trajectory. Okay, as you move on this trajectory, so they keep moving from right to left, just at different slopes, different rates. As you move there, cell functions. We throw different cell cytokines on them and check phosphorylation of transcription factors drop in reduction in how you respond. And when we map this to the Framingham Heart Study, you can basically see that your position on the trajectory, which we call image for this metric of the position of the trajectory, actually matters clinically. We showed in the original paper, we can predict survival up to 7 years ahead when taking all the Framingham Heart Study clinical variables into account, including epigenetic clocks such as the Hovarth clock. So that's kind of where we were in 2019. And just to kind of show you where we've been moving, we've been pushing this much higher resolution. So now we're mapping many more cells of the immune system over time. And rather than looking at chronological age, we've basically moved to thinking about immune aging and this process in individuals because we can map every person and give them an immune age. So this is just a heat map showing you people positioned by their immune age on the x-axis, right? And the frequencies of different immune cells. And you can kind of see there's a cascade here, a language that the immune cells are going through as they communicate with one another. And this is happening in peripheral blood, which we basically always never think of as real immune system, right? It's all about the tissues, the immune cell tissues, and yet we see these processes in blood. This is work by Aviv Barak, a PhD student in the lab. And one of the interesting things is that we started asking is, well, should we be thinking about individual cells, or we should be thinking about this as a system? So a simple way, after a lot of work, we started looking at pairs of different cells, okay? So that, for instance, what you can see here is your naive CD8 T cells, again, in blood, correlation with NK cells. You shouldn't be expecting anything to see in blood, and what you see is that in low immune age, you don't have significant correlation, and in high immune age, you do, okay? So there's some type of conservation like this, and this is just one such paired feature, where somehow there's a conservation of ratios or correlations that's happening, or interactions that's happening in blood. We test this functionally. So for instance, zooming in on T cell subsets, we can see kind of effector memory CD8 T cells. There's shifting in functional markers that suggest that as you move around immune age, you're moving towards terminal state of the CD8s, you're moving towards reduced proliferation, you're moving towards functional impairment. And indeed, when we do functional assays, such as stimulating those cells of individuals, we collect a donor cohort, we give each one of them an image score, and then now we do stimulation assays. What you can see is that, in fact, looking at the ratio of effector memory CD8 T cells to central memory would suggest that they rise, that ratio increases with age, suggesting faster immune responses. And on the other hand, when we look at proliferation, we see reduced responses of that ratio. So your immune age or your immune state, as I measure it in blood, actually matters functionally. And we can do this now kind of in multiple different assays to show that things change. On a more global level, as we say, okay, well, I just showed you two ratios, whether it's central memory, effector memory, or naive CD8 and NK cells, what's happening to the entire architecture of the immune system? And so here, this is again work by Aviv. We looked at basically the correlation coefficient of any kind of triad of 3 cells, right? So interestingly, when you look across immune aging, the number of edges in this network of cells that are interacting doesn't drop significantly. They're, young and old people or young or old immune age people, the interactions exist, right? But we're asking whether they're clustering together. Do you get these tirades of high clustering coefficient or low clustering coefficient, right? And there you see a significant change with immune aging, where there's a reduction that happens in the amount of crosstalk. With aging, that reduction is significant, and yet is still much more than what we'd expect on random, again, suggesting that we can see these observations of how people are immune, kind of what the system is trying to conserve in peripheral blood. And that's really useful as we start thinking about taking these types of biological metrics into the clinic, right? Because I can see this from blood. It's a relatively minimally invasive tissue. The problem is we need scale, and we need to do this in a cheap way in hospital settings. So our original way that we defined immune aging was using a machine called CyTOF. These cost about $1.2 million. They really only exist in research institutes. We're lucky to have one. You can do 30 samples a day on CyTOF, and we can measure 40 proteins simultaneously on single cells. The fact that there are ratios that are dependent on one another, and I just showed you, allowed us to reduce the dimensionality of this and now measure an immune state forget immune cells. I don't care about immune cells anymore. I'm trying to capture an immune state. We can capture image with 8 markers that basically describe this on a flow cytometer, hundreds of different samples simultaneously. It's cheap. It exists in every hematological unit in every hospital. And so we have the original image score and the reduced image score, and they highly correlate. This is work led by Eric Fedman, an MD PhD student in the lab, with Netta and Tim, who are senior staff scientists. So it starts there, and it goes even further, because the immune system is not just about a single data type. There's crosstalk across them. And so what we started doing, using the cohort, this very rich cohort that has multiple data types, is that we started learning proxies for image in gene expression, Affymetrix chip, RNA-seq, whole blood, PBMC, methylation patterns, proteomics, and single cell, and so forth. So we can map any data set that people give us to given metric for immune age and ask how does it play a role. Just to give you a sense, we've been doing this for epigenetics, as I just mentioned. This is work led by Kohav Tal, who's a master's student in the lab. And so we took the entire Framingham-Howard study, measured multiple different epigenetics clocks on them, and then also in what we call image epi, a proxy for image in methylation data. And what we could see is that actually, no matter which clock we're using, image plays and we're modeling clinical variables, image plays a significant role in predicting survival, and in fact, complementary to the epigenetics clocks. And it's maybe not surprising, because once we've done this, and we can do this with 20 CPG sites, there's basically no overlap with the existing clocks in terms of CPG sites. Another thing we've been moving towards making this a clinically actionable biomarker is technical day-to-day stability, before and after fasting, a variety of different things where this is work in progress that we're doing, as well as upscaling this, as I mentioned, for basically getting this protocol up and running for full automation and so forth. So to summarize up until now, what I basically tried to tell you is that we've identified a pattern that exists in all of us in how we change as we go through life and time goes on. That pattern is conserved. It's all like we're driving on a single road. I call this like the first principal component of immune variation, or at least cellular immune variation. And we can now take a blood draw from you and derive an image score that describes where you are on this road right now, right? Now, an interesting thing is that we've been doing this around the world around different cohorts, including a study in Bangladesh where we took 3-year-old kids. In Bangladesh, 3-year-old kids in Palo Alto and older adults in Palo Alto, 3-year-old kids in Bangladesh have an immune age like a 30-year-old in Palo Alto. Okay, so it's really suggesting this is describing the biological or developmental process of the immune system adapting to exposure in life. And another interesting question to ask is, as you travel on this road, and now you took an off-ramp, where would you end up, right? So if we're all cars here traveling at different speeds, and we're in different positions, where would we end up? It's kind of like, are we all going to end up in Rome, or are we going to end up in different positions? So does immune age, does your immune state now, when I challenge you with something, would matter? Now, you already know the answer because I just showed you that it matters for survival, right? But we wanted to get much higher granularity, and particularly we're trying to get payers, insurance companies, to recognize that biological clocks are worthwhile to measure, right? So you need to be very, very specific to identify something that payers care about. Basically, they pay a lot of money for and they get it wrong, right? And knowing a biological clock or this immune aging metric would matter. So we already had a lot of signals suggesting that cardiovascular disease is a place where immune aging is relevant and precedes cardiovascular decline based on standard clinical tests. And we dug head deep into a TAVR study. This is basically a procedure where there's a neurotic valve replacement. It's become now a relatively common procedure in cardiology. You can see here, this is a study in collaboration with Francois Haddad and Holden Maker from Stanford, where we took 96 patients. We had a blood draw, baseline blood draw, right? We have their information. They're going through a TAVR procedure. They're getting all the standard cardiovascular assessments done, echo studies and so forth. And then they have a 1-month follow-up and a 1-year follow-up, okay? And the question is, well, clinicians are making decisions on these people, whether they should go to the procedure, what they think is going to happen and so forth. Would immune aging matter? So if you take all their echo procedure
and you look at this and how people perform, basically at echo at baseline, cardiologists today can describe this as a PCA of echo results, favorable baseline echo results. They know if this happens, the person will actually have a good TAVR procedure. And then they had unfavorable baseline. And if it's unfavorable, you actually don't know what's going to happen. They may end up with ventricular adaptation or maladaptation. And so what we asked is, given if you have an unfavorable echo, can we tell what's going to happen at baseline? So you can see here, AUC of 79.8, being able to, based on your image result at baseline, segregate those individuals into maladaptation or not, which one will end up adapting. And the same thing goes for survival 1 year into the study, into the procedure. So a very pinpointed place where immune aging seems to matter, and having a metric or a biological clock for it is worthwhile. Similarly, we asked about the much more common procedure, which is basically vaccination, and looking at the seasonal flu vaccine, the COVID mRNA vaccine. This is a study with Yuval Dor and Dana Wolf from Hebrew University, led by Mayan Briller and a PhD student in the lab. We vaccinated a cohort of individuals, followed them over time, and asked with neutralizing antibodies, how do they respond to the COVID mRNA vaccine, right? Immune aging may not matter at all, your immune state. For an immunotherapy, it doesn't make sense. It makes a lot of sense that your immune state would actually matter. You can see this on the graph here on the left-hand side, where basically image does actually affect, significantly has an association with the neutralizing antibody, the day 28, versus chronological age does not. When we looked at the entire variance explained in this study, 20% can be explained by your immune age, 10% by your chronological age, and there's still 65% that we need to work on, and we are. Another point is going back, drilling down into this. If your immune age matters, right? So basically that means that you're driving on the road, right? Very nice. And now I gave you a challenge. I gave you a vaccine, and we're already telling you it's going to matter for how much neutralizing antibody you make. But are you driving on the same road when you get, are the same immune components playing a role as you get the stimulation, right? The good response. Now, standard immunological textbooks says, yes, it's the same cells. We know the players, right? But if you look here, this is a very large cohort of influenza patients, 400 patients getting the flu vaccine. And this is a smoothing, kind of a moving average of where they're sorted based on the image. That's the kind of bar. And what are the cells and gene modules that play a role in predicting or in correlating with HAI responses, your titer responses for the flu vaccine? And what you can see here is that different features play a role differently as part of what your immune age is. And this is highly significant in a permutation test, suggesting that as you move in your immune age in an allostatic way, your immune system changes. And I now stimulated you, different players play a role, which opens up a whole new slew of immunology for where, what are cells doing as a function of the microenvironment, if you like. So with that, I'll kind of switch gears. I'll just say, and I hope I convinced you, that immune response matters, and that when you challenge people, they respond, but in some ways, in ways that you wouldn't expect. So to me, this is like playing a soccer game. You know, we think about people scoring goals, they run around and score goals. But old people score goals too when they play soccer, just in different strategies. They do long shots, right? They play a different game. So in the same way, we need to be thinking about immune aging as a process, the developmental process that's changing, and now understand that what the strategy is of the body to actually cope with a different immune system. I'll switch gears to the last two slides, just to give you a sense of how all of this ties to what we're building the Technion. I was here last year. I spoke about the fact that we just launched a Healthy Aging Institute. So now we're up and running, right? So this is a large institute. The Technion basically committed $70 million for this, which I'm lucky enough to direct. It's a fascinating direction of basically taking a technological university and pointing it at the largest health challenge today, right? Which is healthy aging. Basically, we have a cohort that we're building. There's a whole kind of research team, research teams that link to it. And to give you the sense of what we're talking about here and what the Technion can bring, we're talking about research teams that go from cell biology and systems aging, like people like myself who study the immune system, to cognition, biomarkers. Yoni here leads the biomarker theme,
which is all about AI and kind of integrative multimodal directions, all the way to our engineering faculties. People are doing modulation of foods, assistive technologies of robotics, looking at communities and interactions in the community and environments in architecture and civil engineering. Just to give you a sense of the power of putting this under one thing, here's a project that we just launched, involves multiple faculty from the multiple research teams. So Noga Ronen-Noel and myself work on immune aging. Uri Lesmes is a faculty in biotechnology who builds foods, designs foods for older adults. And we're basically launching a project that has how do you modulate immune aging using foods that are actually fitting for those individuals, combined with Merav Aron Gutman, who's an urban sociologist in civil engineering, in architecture, who studies loneliness in the Hadar neighborhood in Haifa, can predict loneliness in people and change urban planning, and we're collecting blood samples from those individuals. So that, I hope, kind of summarizes kind of the activities we're doing at the Technion broadly, and happy to take questions. Thank you.
Thank you very much, Shai. Yeah, we have time for some questions.
I have a question. Great, really fascinating study, and I really hope that you can, use this technology very quickly in human. Now, for this new institute, 70 million institute, so are you going to recruit new staff, or you just use the currently existing Technion scientists?
We're both recruiting new faculty. So first of all, powering, I will say, and this is how we think about this at Technion. So first of all, our president decided that human health is a strategic direction that Technion needs to push, which is a big decision for the Technion. It means a lot of Technion money is going into human health. Within this, we decided that the topmost thing is healthy aging. And the institute basically launched. It both includes recruiting new faculty members, so we're looking. Talk to me if you're interested.
Within Israel?
Within Israel right now. And empowering a lot of synergism between Technion departments and so forth that normally, people are used to their lab. And we're trying to basically put people together across the campus to take, kind of make real life impact challenge.
That's very interesting though. Let's make really the longevity nation, for Israel to be ahead of other countries, right? Hopefully.
Thank you.
Thank you.
So I guess it wouldn't be crazy to preserve my young state of immune system and re-inject my own blood 30 years later. What would you say happens?
Well, I'm not a physician, so I cannot recommend it. But preserving blood is easy. I would go for it. While it's waiting, I'll say we already know that dependent on the condition, it may change. So we're looking, for instance, at bone marrow transplant. We believe an old immune age is a benefit for a donor, for a recipient to get an old immune age donor. Results coming next year in this talk, if Ilya invites me.
Comment on this Blind Johnson story. What do you think? Everybody is fascinated about that.
I subscribe to a different camp. Yes, please.
Thank you. So you did mention testing your biological age, knowing where you're at. And is there a specific age that you should shoot for, which is optimal for maintenance? For example, regardless of how old you are, as long as your biological age is at a specific number, it's good for maintenance long term. So is there like a specific age you should shoot for when it comes to biological age?
I think it's an interesting question, a fascinating question. I don't think we're close to having an answer. As I just was saying to Leonid, it's quite clear to us that the condition changes the type of immune age you want, that would be of preference. So if you're living, for instance, in an infectious environment that has a lot of infections, you could think about in the young immune age as being a good thing, right? If you're getting a bone marrow transplant, an old immune age would be a good thing. If you're living at the Bangladeshi data would suggest that if you're living in an area that's really high, kind of pathogen load, the immune system adapts to having an older immune age because you just can't, you can't continuously have such an aggressive immune system attacking kind of a bystander effect on the host. So I think the answer is it's going to take us some time. That's a great question.
Yeah, thank you. Just goes to show how resilient our immune system is when it comes to that. You adapt to your environment pretty much.
Right. So what we are doing, just to give a sense of this, is I strongly believe it relates to resilience in that sense. And we're trying to use, for instance, fitness to describe what are the things that are changing in the immune system that are, our lifestyle. You can change what lifestyle versus not, right? What are the things that are basically maladaptations versus adaptations? And I think once we start decoupling them, the picture is going to be much. So again,
Awesome.
Ilya, I'm hoping Ilya will invite me next year. And,
Great.
Thank you very much, Shai. I think because of the organizational concerns, this is the end of this session, and Catherine will give her talk in the next session.